Hi. For anyone who may not know me, my name is Eric Weiss, and I'm a sixth-year PhD student in Julia Saltz's lab here at Rice University. And today, as part of my dissertation defense, I'll be discussing what exactly I've been up to for going on 6 years now. Throughout my time here at Rice, I've sought to answer a myriad of questions addressing the causes and consequences of social group structure and how these factors shape how we might expect sociality and the structure of social groups to evolve. So it should come as no surprise to many of us that the social interactions an individual engages in can have dramatic influences on an individual's fitness. Social interactions can influence everything from how individuals acquire and use social information, to how individuals collectively forage, to individuals' propensity for contracting and spreading diseases and their opportunities for mating. Because social interactions are so important to fitness and a major target of selection, understanding the quantitative genetic basis of social traits is a necessary step toward understanding how social behaviors evolve. After all, in order for evolution to shape variation across generations, we have to know more than just how natural selection acts on phenotypic variation. For there to be phenotypic response to selection in the next generation, quantitative genetic variation, meaning variation that's explained by the cumulative effect of an individual's genotype, as opposed to a singular or a few genes, has to be part of what generates that phenotypic variation, as opposed to phenotypic variation being entirely due to effects of the environment. However, for social traits, things get a lot more complicated because part of the environment that individuals experience and that shapes their phenotypes actually has a genetic component to it. And that is the social environment that individuals experience. So let's take this second individual here. This individual, whose phenotype is also dictated by its genes and the environment, can dictate the expression of a phenotype in this focal individual. When the genes of other individuals end up influencing your own phenotype, these indirect genetic effects, or IGEs, are an additional component of quantitative genetic variation that can shape social traits. Additionally, the presence or actions of the second individual can influence the fitness of our focal individual, and the degree to which it does so is a force called social selection that, depending on the genetic variances and covariances between the interacting individuals, can lead to interesting evolutionary outcomes, including the social environment itself evolving. Social selection is also an important process, as its outcomes may run counterintuitive to what you'd expect via natural selection alone. For instance, social selection may act in a way that strengthens the force of natural selection, but can also act counter to what we'd expect to see via natural selection alone. But even with these added layers of complexity, these processes are still a simplified depiction of what's actually occurring in nature. Because rarely are individuals interacting with just one individual who can shape their social phenotypes and fitness. Individuals frequently interact with dozens or more conspecifics, each of whose behaviors can shape not only a focal individual, but each other simultaneously. So it's not just that the social interactions that individuals engage in is important, it's how these interactions play out within social groups of individuals. And similar to how social interactions are a key component of individuals' life histories, how individuals are nested within entire social groups of interacting individuals can have far-reaching consequences for many aspects of social group members' behaviors and fitness. So the full picture looks less like this and more like this, where multiple individuals not only shape a single individual's phenotypes and fitness, but all of these individuals can interact and influence each other's behaviors and fitness. And we end up in a situation where there are so many interacting effects that it can be extremely challenging to make sense of it all. Luckily, one of the great tools we have at our disposal to quantify how individuals are nested within a larger social group of socially interacting individuals is social network analysis. In brief, a social network can be described by points or nodes representing individuals, and the lines connecting them, or edges, represent interactions between any given pair of individuals. What network analysis allows us to accomplish is not only quantifying and describing direct social interactions, exemplified here with the red individual's direct social partners, but it also allows us to describe indirect social interactions as well. Shown here is how the red individual is indirectly connected to others. A key example of the importance of indirect social interactions can be exemplified by something I'm sure we've all experienced over the past year, and which are COVID bubbles. Your risk of contracting disease depends not only on your direct social interactions, but indirect social interactions occurring between people you're directly and indirectly connected to. And it's not only disease that makes the position you hold in your network important. The structure of social networks and the positions of individuals within them has similarly been implicated to impact many components of individuals' fitness and selection on these network positions. Which brings us back to here. In order to understand how traits respond to selection and evolve, we have to understand the quantitative genetic basis for these traits. And for social traits, like how entire networks of social interactions are structured, we have to replicate and control for the genotypes of all individuals involved in a social group. Of the few studies that have sought to estimate the genetic basis or heritability of social network position, basically all have done so by measuring focal individuals of known relatedness or genotype, such as this red individual, against a backdrop of uncontrolled individuals of unknown genotypes. If you try to measure the network position of focal individuals of known relatedness or genotype against a backdrop of uncontrolled individuals of unknown genotypes, even if the focal individual behaves exactly the same across social group contexts, this can induce a lot of variation in network positions measured for that particular individual. So this leaves a big open question about how quantitative genetic variation, including both direct and indirect genetic effects, shapes the structure of social groups. And with everything we know about how network structure has profound fitness consequences for individuals, understanding the quantitative genetic basis of social processes that incorporate both direct and indirect social interactions allows us to better understand how social groups can respond to selection and evolve. Subsequently, while we know social selection and individuals' network positions are vitally important for fitness in many systems, we know little about how these processes act on genetic variation in social network structure. Studying both genetic variation and selection simultaneously gives us a fuller picture of all the components necessary for evolution to proceed. Lastly, while both of these processes are critical for understanding how social group structure evolve, it's important to note that genetic variation and selection themselves can be variable, with numerous processes affecting them. Understanding how both genetic variation and selection depends on the context where individuals find themselves can be just as important in shaping the evolution of traits, including complex social phenotypes. For example, we typically think about how selection and how it reduces phenotypic variation, but context dependence is one of the many ways that evolution can actually maintain variation, which can help explain why so much diversity persists in nature. To unpack a little bit more about what I mean by this, imagine a situation where you have two genotypes, genotype 1 and genotype 2, and two environments, environment A and B. In environment A, genotype 2, the red line, has larger value of some phenotype compared to genotype 1. But in environment B, genotype 1 has a larger phenotype compared to genotype 2. If we pair this with how selection acts on this phenotype, where larger values lead to higher fitness no matter the environment, we'd see that genotype 2 is favored by selection in environment A because its phenotype is larger, but genotype 1 would be favored in environment B. Thus, interactions between genotypes and their environment, termed genotype by environment interactions, or G by E, can maintain genetic variation when the environment is variable and selection is consistent. Additionally, if the effects of genotype are consistent across environments, shown here, but how selection acts on variation varies depending on the environment, shown here, then context-dependent selection can maintain genetic variation. In our hypothetical example, genotype 1 would have higher fitness in environment A, the green genotype, but because genotype 2's phenotype is overall lower than genotype 1, it would have higher fitness in environment B, the brown environment. Because which genotype benefits depends on how selection acts in a given environment, genetic variation can be maintained. And understanding how context-dependent genetic variation and selection act on social phenotypes can further round out our understanding of what shapes the vast diversity of social structures we see in nature. And as the topic of the final question I'll be addressing today, which is how selection and quantitative genetic variation is shaped by context dependence. Okay, now answering these questions together can shed light on how the causes and consequences of variation shape social group structure. To answer these questions, we need a system where, as I mentioned earlier, we can control and replicate the genotypes of all individuals within the social environment. Ideally, we'd be able to track individuals' fitness throughout their lives to understand how selection acts on variation in individuals' network positions. And we should be able to easily manipulate components of the environment that individuals experience to assess how context dependence shapes genetic variation and selection. Which brings me to flies, or Drosophila melanogaster. Using flies allows us to control the genotypes of many individuals using dozens of inbred lines derived from a wild population. They have easily tractable lifespans of around 70 days, so we can measure components of fitness throughout their entire lives. And we can subject flies to many different environmental contexts to see how quantitative genetic variation and selection shape the structure of social networks. Now, if you think to yourself, hold up, wait, you're studying social group structure using flies? Well, then I've got really great news for you, because flies are actually an incredible system for addressing questions about behavior and social group structure. Flies engage in all sorts of cool behavioral interactions. They fight with each other, like you can see with these two flies throwing down together. They engage in stereotyped courtship rituals. But at the group level, flies have also been shown to non-randomly choose and form social groups, as well as form non-random social interactions, where flies preferentially associate or disassociate with one another. This leads me to two experiments I'll be discussing today, which seeks to address how all of these questions are affected by context from two different angles. First, I'll be addressing how genotypic variation and selection shapes the structure of replicate social groups, and how these processes vary across different nutritional contexts that individuals experience. Flies are a great ecologically relevant system for addressing how nutritional context affects genetic variation and selection, as social groups of flies live, they eat, they mate, they lay eggs on nutritional environments that are highly heterogeneous, both in terms of what foods they eat and the quality of the food patches that they're on. And secondly, I'll be discussing results from an experiment where we manipulated the social context individuals experienced. Because as I mentioned previously, individuals vary in the social environments they choose to experience. So measuring how they're affected by different social environments can help us understand how selection shapes this variation. For our first experiment, we were primarily interested in how direct genetic effects shape individuals' positions within their networks of interactions, while controlling for the genotypic identities of all other individuals in their social groups. As such, we created replicate social groups by taking 40 replicate inbred lines of flies, shown here as 40 different colored flies, and performing 20 mating crosses between them. By crossing these homozygous genotypes together, we create 20 individuals that are heterozygous at every loci that their parents differ at. Since the parents contribute the exact same genetic material to their offspring every time a cross occurs, we can continuously create these heterozygous genotypes to use for experiments. There are a couple reasons we do this. One is to help alleviate the potential effects of inbreeding, and two is to create individuals that are more representative of what you would expect to find in a wild population. But we can do this over and over again, as many times as we'd like, to replicate individual heterozygous genotypes. Of the 20 heterozygous genotypes we created, we used virgin females from 10 of the crosses and virgin males from the other 10 for a 50-50 sex ratio. We painted a unique color on the back of each individual to tell which genotype was which. And we combined these 20 individuals into a social group that we replicated about 100 times. Each social group was then put in a different nutritional environment that varied in 2 dimensions of nutrition. First, either how calorically concentrated the food was, ranging from very dilute to very concentrated food, or food that had the same amount of calories, but varied in whether the calories were derived more from protein or carbohydrates, which we manipulated by changing the yeast and sugar ratios in their food. And both of these dimensions of nutrition have been shown to impact many components of flies' behaviors and fitness. To quantify network structure, social groups were video recorded over the course of 2 days. Then, using motion tracking software, we were able to output the physical locations and tracks of every fly throughout every frame of a video. And using this, we wrote a program that quantified the number of social interactions occurring between every pairwise combination of individuals, using social interaction criteria based on things like distance between individuals, the angles they were facing other flies, and how long these interactions took place. We quantified 56 replicate social networks encompassing 1,120 individuals, which allowed us to estimate the positioning of every individual within their networks. And lastly, to address how selection acts on genetic variation in network position, we measured 4 components of individuals' fitness that have been shown to be influenced by individuals' behaviors and nutritional context. For males, we measured their total mating success and their latency to mate in the first 2 hours after social groups were established. For females, after social groups were allowed to interact for 2 days, each female was removed and placed in an individual vial with a standard food. And we counted all offspring that emerged throughout a female's life, as well as their lifespans. Okay, so together, this allows us to address our questions about how quantitative genetic variation and selection act on network structure, as well as how these processes vary across different nutritional environments. Okay, so for addressing how quantitative genetic variation shapes the structure of individuals' network positions, we specifically looked at 5 measures of network position that either described individuals' direct or a combination of direct and indirect social interactions. In strength, or the number of social interactions individuals receive from others. Out strength, the number of social interactions individuals initiate to other individuals. Eigenvector centrality, which is basically a measure of how important an individual is to the structure of its social group, given the strength of both its direct connections and its indirect social connections. Betweenness centrality, which quantifies how much an individual serves as a bridge between indirectly connected individuals in its network. And clustering coefficient, which describes the degree to which individuals you are socially connected to are also connected to each other. So it can be thought of as a measure of how cliquish individuals are. These measures of network position are the most commonly studied and described components of network structure, and all have been shown to be influential to components of individuals' fitness in many different systems. Using a combination of generalized linear mixed models and permutation tests to account for the non-independence of network data, we were able to test how an individual's genotype and sex influenced each measure of network position. Okay, so each of the figures below each network position parameter shows our results for how 20 genotypes varied in each network position. On the y-axis for each graph, we have the values for each of our 5 network position parameters. And along the x-axis for each graph, we have 20 box plots, one for each of the 20 genotypes in our social groups. We found that genotypic variation significantly predicted all 5 measures of network position. The proportion of variation explained by genotypic effects, defined as broad sense heritability, ranged from about 2.5% up to almost 17%, depending on the network position parameter. Additionally, we found significant sex differences in 4 out of the 5 network positions measured. Unsurprisingly, females, the yellow box plots, were found to receive more social interactions, while males, the blue box plots, initiated more social interactions. Females were generally more central to the structure of their social groups. And while we didn't find any sex differences in how individuals served as a bridge between indirectly connected individuals, we did find that females were much more cliquish and engaged in more clustering compared to males. So now, understanding that these network positions have a quantitative genetic basis when the genotypes of everyone in a social group is controlled for, we can ask how this variation is affected by the environment. We found that genotypic variation in network positions was largely consistent across nutritional contexts. However, while we found little evidence of genotype by environment interactions for network positions, we did find evidence of sex by environment interactions for network positions. Specifically, while females, the yellow box plots, and males, the dark blue box plots, initiated similar amounts of social interactions, so there's out strength on the y-axis, in environments that were either high in protein or calorically dilute, shown on the x-axis, They diverged on environments that were either high carb or calorically concentrated, where males ended up initiating more social interactions than females. Similarly, while females were consistently more cliquish and clustered more, shown on the y-axis of the bottom two graphs, they became more cliquish in nutritional contexts of increasing carbohydrate and increasing caloric concentrations, while males became less cliquish in high carb and calorically concentrated nutritional contexts. In addressing how selection acted on genotypic variation, we didn't find any evidence of selection acting on network positions in females. We did, however, find selection acting on measures of network positions in males. Males who engaged in more social interactions and were more central to group structure had lower mating success overall. But most interestingly, these effects of selection differed depending on the nutritional context males mated on. More specifically, males who engaged in more social interactions or were more central to group structure, shown on the x-axis, had higher mating success, shown on the y-axis, but only on nutritional environments that were high in carbohydrates, the dark gray line in each figure panel. But in nutritional contexts that were higher in protein, which is the light red line in each panel, engaging in more social interactions and being more central to group structure correlated with lower mating success. A similar pattern held for nutritional contexts that varied in their caloric concentration, where males who engaged in more social interactions and were more central to group structure had higher fitness only in nutritional contexts of calorically concentrated food, again, the dark gray line, with the opposite relationship occurring in calorically dilute nutritional contexts, the light teal line. These same general results were also found for effects of males' latency to mate, where engaging in more social interactions correlated with faster mating in high carb and high calorie contexts, with the opposite trend occurring in high protein and low calorie nutritional contexts. So if we tie all this together and go back to the main questions we set out to address, we find that measures of social network position that incorporate both direct and indirect interactions are shaped by individuals' own genotype and are heritable. Selection acts on this genetic variation. However, the strength and direction of selection depends on the context social groups experience. While these findings are some of the most robust evidence we have that the structure of social groups can evolve, a key piece of unknown information remains, which is why we found network structure to be heritable in replicate social groups. Individuals often experience variable social contexts with different individuals. After all, we know that flies and individuals of many species shape the social environments they're in many ways. So studying how the genotypic composition of your social environment impacts network structure and selection may be just as important for understanding the evolution of social group structure. Which leads me to the second experiment I'll be discussing, in which we manipulated the social context and genotypic composition of social groups that individuals were a part of. We could then ask how indirect genetic effects of individual social partners influenced their network positions, as well as how selection varies depending on individual social context. So to create social groups of variable genotypic compositions, we chose 3 heterozygous genotypes of female flies that, based on data from our first experiment, varied in how much they influenced the structure of their networks, i.e. their eigenvector centrality. We chose to manipulate only one sex, since varying both sexes would introduce confounding variation about which specific genotypic and sex combinations influence group structure, and this wasn't the goal of our study. We also chose to manipulate females, since we'd previously demonstrated females to be more influential to the structure of their groups. We then paired each of these focal genotypes with a group of 9 stimulus individuals of the other genotypes. Sorry, computer's being a little bit slow because the PowerPoint's big. So we did this in a full factorial design, but excluding pairings where the same genotype was paired with the same genotype. The oddball or less common genotype in each group we'll refer to as the focal individual of the group. And individuals who are one of 9 individuals of the same more common genotype in the group are referred to as the stimulus individuals. Okay, we then added 10 males of a standardized genotype to each group for a 50-50 sex ratio. There we go. And replicated each of these social contexts about 20 times for a total of almost 120 social groups. Okay, we measured the structure of social networks the same as the first experiment I discussed, where we generated networks from videos taken of the groups. And we also measured the same metrics of fitness I previously mentioned: mating success and latency to mate for males, and offspring production and lifespan for females. And together, this allows us to address not only how direct genetic effects shape individuals' positions within their networks, but we can address how indirect genetic effects of social partners shape network structure. And since we manipulated both singular focal individuals' genotypes and the genotype of groups of stimulus individuals, we can address not only how variable social groups impact individuals, but how the presence of a single individual can shape the social network structure of everyone else in the group. This setup allows us to address two different components of context-dependent selection as well. We can address how selection on network positions varies depending on the social context individuals are paired with, but we can also address an additional component of context-dependent selection, which is frequency-dependent selection. Because genotypes in a group were either on their own, the focal genotype, or one of 9 individuals of the same genotype, the stimulus genotype, we can address how individuals' genotypic frequency in a group shapes their fitness, as well as whether frequency-dependent selection varies depending on indirect genetic effects of the genotype individuals are paired with. Okay. All right. Okay, so because females were the only genotype that varied in our social groups, we could address how direct genetic effects impacted their network positions, but we couldn't do that for males since males all had the same genotype. We also analyzed the effect of singular focal genotypes and the group of stimulus genotypes separately, since an individual's own genotype correlated with either the focal or stimulus genotype treatments of the group. Okay. So for singular focal individuals within the group, shown along the x-axis, we found that an individual's own genotype only influenced 2 measures of network position. That is, how central individuals were to the structure of their groups, their eigenvector centrality, shown on the y-axis, and how individuals served as a bridge between indirectly connected individuals, i.e. their betweenness centrality. For the stimulus female genotypes, shown along the bottom, we similarly found that an individual's own genotype influenced both how central individuals were to the structure of their networks, as well as how individuals served as a bridge between indirectly connected individuals within their networks, but also how cliquish individuals were, also known as their clustering coefficient, was influenced by individuals' own genotype. One of the interesting things about these results is that while we might expect an individual's own genotype to be more influential of its direct social interactions, like in-strength and out-strength, these are the only 2 measures of network position that we didn't find evidence of direct genetic effects. Yet measures of network position that incorporate indirect interactions were influenced by direct genetic effects. This nicely sets up our results for the additional component of quantitative genetic variation we tested, which is how indirect genetic effects shaped individuals' positions within their networks. Okay, so we found that focal females' network positions, i.e. the singular genotype within a group, was impacted by which stimulus genotype these focal females were paired with. More specifically, how focal females served as a bridge between indirectly connected individuals, their betweenness centrality on the y-axis, varied depending on whether focal females were paired with a group of stimulus individuals of either genotypes A, B, or C, shown on the x-axis. Also, we found the same pattern for how cliquish focal individuals were. And which varied depending on whether the focal genotypes were paired with either genotype A, B, or C. While we found indirect genetic effects of the stimulus genotypes on focal genotypes and network positions, we didn't find the reverse effect of focal genotypes influencing the network positions of stimulus genotypes, shown along the bottom graphs. This perhaps isn't too surprising, however, as it's likely much harder for a single individual to shape the structure of their network compared to a group of individuals influencing network structure. Also, while we couldn't test for direct genetic effects in males because males' genotypes didn't vary, we could test for indirect genetic effects of females on males' network positions, where we also found that stimulus genotype males were paired with shaped how central males were to the structure of their social group. Okay, so knowing that individuals' positions within their networks can be shaped by both direct and indirect genetic effects, we then asked how selection acts on this variation across different social contexts. We didn't find evidence of network position impacting males' mating success or latency to mate, nor did we find selection operating on females' lifetime offspring production. We did, however, find selection operating on measures of network position, shown on the x-axis, via our fitness component of females' lifespan, shown on the y-axis. And these selection gradients varied depending on indirect genetic effects of which genotype females were paired with. For example, when individuals were paired with genotype B, the yellow genotype, then engaging in more social interactions, either in strength or out strength, and being more central to group structure, eigenvector centrality, had little to maybe a slight increased effect on individuals' lifespan. Yet if individuals were paired with genotype A, the blue genotype, then engaging in more social interactions and being central to group structure were costly for females' lifespans. Interestingly, the network positions we found to be under selection are ones that encapsulate more information about direct social interactions. Yet the data from this same experiment showed that network positions that encompassed more indirect interactions were more likely to have a quantitative genetic basis. That being said, the other evidence we've accumulated shows that these network positions can be heritable depending on different contexts. But even aside from estimating selection on network structure, we were also able to test an additional component of context-dependent selection. That is, since the female genotypes in our groups were either 1 of 20 individuals, the focal genotype, or 9 of 20 individuals, the stimulus genotype, we could address how frequency-dependent selection was acting and whether being the rare or common genotype impacted females' fitness. And subsequently, not only did we find evidence of positive frequency-dependent selection, meaning that being a part of the more common stimulus genotype, shown on the x-axis, benefited both components of females' fitness, shown on the y-axis, lifespan and lifetime offspring production. We also found that this frequency-dependent selection depended on the social context individuals experienced. For example, being the more common genotype increased how long females lived. But if a female was also paired with genotype A, the blue genotype, this effect was much more pronounced. Typically, positive frequency-dependent selection operates to reduce genetic variation, where less common genotypes are selected out of the population in lieu of more common genotypes. However, because we found that frequency-dependent selection depends on the social context individuals experience, this context-dependent selection can serve to maintain genetic variation. And so in reviewing these results, we find that an individual's position in a network is not just shaped by the direct social interactions of its own genotype, but indirect genetic effects of social group mates can be just as influential to how individuals are structured within their social networks. Furthermore, how selection operates on the structure of social groups is strongly dependent on the genotypic makeup and composition of social groups. So it's critical to understand not only how selection is acting on the structure of social groups, but that the actual makeup of individuals within these groups likely shapes both how they are structured and selection on structure. Okay, so when we take a step back and think about what all of these processes mean for the evolution of social group structure, the main points to take away are that quantitative genetic variation can shape the structure of social groups. And it's extremely important to know that quantitative genetic variation in complex social traits is derived from both direct and indirect genetic effects. When we compare the two studies I discussed, it's notable that we found evidence of direct genetic effects on all the social network positions we tested, when each social group was a replicate containing the same set of 20 genotypes. But in the social context experiment, we found that only measures of network position that encompassed indirect social interactions had a direct genetic component. This could be because variation in the composition of social groups obscures direct genetic variation. Alternatively, in our experiment where each replicate social group contained the same set of 20 genotypes, It's possible that consistent indirect genetic effects may have been driving our findings of direct genetic effects. Taken together, estimating the quantitative genetic basis and evolutionary potential of social group structure without understanding the genotypic contributions of everyone in a social group risks missing crucial components of variation. Granted, this can't always be done in many systems, but hopefully future work will continue to address this, as well as develop better ways of linking quantitative genetic theory and network analysis. And the second major point I'd like everyone to take away is that when estimating selection on social group structure, context matters. If we go back to one of the slides from earlier about how context-dependent genetic variation and selection can serve as mechanisms that maintain variation in phenotypes, we'll see that while we found only little evidence of genotype by environment interactions, context-dependent selection was one of the most consistent themes that we found throughout this work. And context-dependent selection, combined with quantitative genetic effects, can be a major mechanism driving the vast diversity of social group structures we see in nature. This is important because studies of context-dependent selection are extremely rare for studies of behavior, and even rarer for studies of social network and group structure. Yet understanding the evolutionary trajectories of social group structure may be entirely reliant on the given context in which selection takes place. For example, our findings of males having higher fitness when they interacted more or were more central to group structure might lead us to expect groups to become more male-centered or tightly connected over time. But the fact that this was only true in environments that were calorically or carbohydrate rich, with the opposite pattern occurring in nutritionally poor environments, may lead us to expect the opposite. So studying selection in only one setting may lead to spurious inferences about how social group structure will evolve. Additionally, it's important to note that measures of network position are all derived from the same underlying network of social interactions. So selection on one is likely to change the underlying structure of the network, allowing other components of the social environment to evolve consequently. With selection acting on group structure, every new generation will have a new set of individuals and a new social context upon which selection can act. And with our findings, the strength and direction of selection depends on the social context. Evolutionary feedbacks between selection and social group structure are likely to further shape variation in group structure. And further studies would be wise to investigate these processes over multiple generations. Doing so can take us even further into understanding what shapes the vast array of social structures evident in nature. And lastly, if you come away from today thinking, this is really cool, but I don't study behavior or social groups, then it's important to note that these principles are important at many levels of biological organization. Networks of interactions occur at pretty much every level of biological organization, from gene and protein networks up to ecosystems of multi-species interactions. So understanding not only how individual genes, individuals, or species affect each other is important, we also need to understand that these entities are reciprocally affected by one another, and that any given entity that interacts with other parts of its network can be shaped by the presence of other genes, individuals, or species in their genomes, groups, or ecosystems. And with that, I hope you've come away learning something new and cool about social group structure. And now is a moment I've really been looking forward to, where I can give a big old happy thank you to a lot of people. First and foremost, Julia and all of my lab mates in the Salts lab have been absolutely amazing over the years. Being the first grad student in a lab comes with so many new and exciting challenges, and I know it's been extremely gratifying to be part of the founding of the Salts lab and to work with all of y'all over the years. And equally, or even more important than the postgraduates I've worked with, are all of the students I've worked with during my time here at Rice. I'm seeing all of y'all go on to succeed in college, careers, graduate school, not to mention just becoming better scientists. It's easily been the most gratifying part of grad school for me. And I'd say seeing y'all succeed is even more rewarding than the research part of grad school, to be honest. So thank all of y'all, not only for your contributions to my experiments and the independent projects that y'all have embarked upon, but making me a better mentor along the way and making grad school even more fulfilling. Grad school has been tough, no doubt. And as pretty much all of us know at this point, a lot of times outside of grad school can be even tougher. These past 6 years have been the hardest of my life. I lost a grandfather, a grandmother. My dad died in my first year of grad school. My dog Onyx died. And even I almost died a couple times between a car accident and rupturing my appendix. And that's not even to mention all the horrible things that have impacted all of us over the past 6 years, between constant and persistent threats to our civil liberties, Hurricane Harvey, which upended many of our lives here in Houston, and of course, the worst pandemic in over 100 years. These past 6 years have really sucked, to put it lightly. But despite all the chaos, the past 6 years have also brought about some of the greatest times of my life. I met Beth, we got engaged, we got married, we adopted a new dog together. And also, with all these ups and downs of my time here at Rice, I can't imagine a better or more supportive cohort of people to fight through it with. All the grad students here at Rice have easily made my decision to come here one of the best I've ever made in my life. And last but certainly not least, I'd like to thank my dissertation committee, the biosciences department, and Rice University as a whole, the Rice Counseling Center, multiple funding sources, including the Society for the Study of Evolution, the Animal Behavior Society, and the National Science Foundation, without whom I certainly wouldn't be here today to share my work with y'all. And with that, thank you all for listening, and I'd love to take any questions y'all have. Thank you.